Hello, viewers, and welcome to my Phlebotomy study log. In this video, we're going over some important questions for the chapter on special non-blood collection procedures. Before I reveal the answer for each question, I encourage you to pause the video and think about it yourself. Let's get started. Number 1. An 8-hour urine specimen is typically collected. The correct answer is in the morning. 2. Which urine specimen is most commonly used to determine pregnancy? The answer is first morning. The sweat electrolytes or chloride test is typically used to screen for cystic fibrosis. Number 4. A urine specimen is labeled after collection. 5. A catheterized urine sample may be collected when a midstream clean cut specimen is not obtainable. When the patient is unable to produce a specimen independently by the physician, nurse, or medical assistant. A 72 hours stool specimen is collected to determine fat quantities. A semen analysis must be delivered to the laboratory within 30 minutes of collection. 8. The following body fluids are always treated as stud specimens except for urine. 9. The test used to diagnose whooping cough, croup, and pneumonia is the NP culture. 10. The following specimens are always collected by a physician except for urine specimen. 11. Define a random urine specimen and what it is used to screen for. Random urine specimens can be collected at any time. They are used to screen for obvious abnormalities in the concentration of proteins, glucose, and other significant constituents of urine. 12. Explain the differences between a first morning specimen and a time specimen. A first morning specimen is collected after the patient wakes up. It is a very concentrated specimen. A time specimen is collected over a 24 period to provide a single large specimen. Typically, the first morning sample is discarded in a time specimen. Describe the procedure for collecting a midstream clean cut specimen. To collect a midstream clean cut specimen, the patient should cleanse the area surrounding the urethra. Begin voiding into the toilet. Bring the container into the urine stream until sufficient urine has been collected, and then void the remainder of the urine into the toilet. Explain why fecal specimens may be collected. Fecal specimens are collected to look for intestinal infection and screen for colorectal cancer. Explain why semen specimens may be collected. Semen specimens are collected to determine whether viable sperm are present, either to test for fertility or to assess the success of a vasectomy. Semen may also be collected as a forensic specimen from a rape victim. In CSF collections, how many tubes are collected and which departments receive which tubes? In CSF collections, three tubes are collected. Tube 1 is delivered to the chemistry laboratory. Tubes 2 and 3 are delivered to the microbiology and hematology laboratories. Which department receives which tube is set by the policies of the institution. Explain why amniotic fluid may be collected. Amniotic fluid is collected to analyze the presence of certain genetic disorders, such as Down syndrome. It can be analyzed for lipid to indicate lung development, bilirubin associated with hemolytic disease, and proteins associated with other abnormalities such as spina bifida. Explain the difference between a throat culture and an NP culture. A throat culture is collected from the back of the mouth around the tonsils and uvula with a swab. A nasopharyngeal culture is collected through the nose. Define the body areas where the following body fluids are found and indicate whether the phlebotomist, the nurse, or the physician will be responsible for obtaining the specimen. A. Pleural fluid. B. Synovial fluid. C. CSF. Pleural fluid is found in the lungs and thoracic cavity. Synovial fluid is found in the knee joint. Cerebrospinal fluid is found in the brain and spinal cord. All of these specimens are collected by a physician only. Give some of the findings that a 24-hour urine specimen will yield and at least one method of yielding the results. A 24-hour collection may detect low levels of certain proteins and hormones. Explain two methods used to induce sweating in the SE test and give the equipment used to collect, preserve, and process the specimen in the laboratory. The methods for inducing sweat in an SE test use iontophoresis and the drug pilocarpine. Sweat is collected on a sterile filter paper, which must be covered with paraffin and handled with forceps or sterile gloves. The chloride level is obtained via weight and analysis of electric charge.